Bace1-dependent amyloid processing regulates hypothalamic leptin sensitivity in obese mice. Obesity levels are at a record high. A study conducted in 2018 by the World Health Organization indicates that 28% of UK adults are classified as clinically obese. This comes with major strains on the NHS costing billions of pounds each year. The increasingly easy access to a calorie-dense diet and the reduced amount of physical activity required in daily life could be to blame. However, biological factors could also come into play. Leptin is a cell-signaling hormone released from adipose tissue, and its primary target is the hypothalamus. Leptin plays an important role in regulating appetite, food intake, energy expenditure, and body weight. When high levels of leptin are present in the body, it signals to your brain that you have plenty of fat stored, repressing appetite. Conversely, when the body has low leptin levels, it signals to your brain that you should eat. In an obese individual's body, many leptin molecules are released due to high mass of fat cells. This is called hyperleptinemia. This results in the decrease in sensitivity of leptin receptors in the brain. Resistance to leptin causes a disruption to the feedback loop, which can lead to uncontrolled food intake and weight gain. Therefore, a compound able to reverse or prevent leptin resistance in obese individuals could hopefully aid in the normalization of weight and improve glucose homeostasis. Bace1 is a cleaving enzyme catalyzing the rate-limiting step of amyloid beta production from amyloid precursor protein. This amyloid beta protein plays an essential role in neural growth and repair. The protein in high levels is known to form plaques, thought to contribute to neurodegeneration in Alzheimer's disease. High levels of these plaques also increase central endoplasmic reticulum stress and neuroinflammation, theorized to cause reduction in leptin activity. Bace1 works by cleaving the amyloid precursor protein found on many tissues, including the brain and spinal cord. This then allows Y secretase to cleave the remaining membrane-bound portion, releasing the amyloid beta. This protein can then go on to aggregate into plaques, which negatively affect leptin signaling. We looked at the paper, Bace1-dependent amyloid processing regulates hypothalamic leptin sensitivity in obese mice, to investigate this process. In the study, mouse models were used to implicate the action of Bace1 in subsequent beta amyloid production in the development of leptin resistance. The study hypothesized that consumption of chronic high-fat diet sufficient to cause obesity in mice increases neuronal Bace1 activity and amyloid beta peptide levels, resulting in modification of the processes that act to limit hypothalamic leptin sensitivity, resulting in body weight gain and loss of glucose homeostasis. Leptin, when released, modifies the hypothalamic function by controlling the transcription of key regulatory proteins. Leptin binds to the leptin receptors. This results in the recruitment and activation of JAK2. JAK2 goes on to phosphorylate the tyrosine residues found in the cytoplasmic domains of the leptin receptors. These tyrosine residues act as a docking site for cytoplasmic adapters such as STAT3. The phosphorylated STAT3 then goes on to regulate gene expression. As shown in the study, amyloid beta production increased after 20 weeks of high-fat diet in mice, showing that obesity elevates hypothalamic Bace1 activity. These high levels of amyloid beta have been implicated as an amplifier of central ER stress and neuroinflammation. As shown in the study, high levels of inflammatory cytokine IL-1 beta are more present in the high-fat diet mice compared to mice with no Bace1. A high-fat diet leads to high Bace1 activity, causing high levels of amyloid beta plaques. High levels of these plaques lead to increased neuroinflammation and ER stress, which in turn negatively affects leptin sensitivity. In cases that Bace1 is inhibited using the M3 inhibitor, metabolism is only altered in diet-induced obesity mice, not in mice lacking leptin receptors. This indicates that the Bace1 inhibitor requires leptin and functional leptin receptors to work. We spoke to Alison McNally, who was involved with the paper, asking her further questions relating to the study.
So Bace1 it's a kind of promiscuous protein. We still don't really know what its actual ligand is, so it cleaves a lot of things. It's always been associated with Alzheimer's disease just because it cleaves APP, but this is kind of a new aspect. We noticed it was mainly from the mouse models that we noticed they just don't get fat, so it's like, what's going on?
Because it's like Bace1 inhibitors, I think they're under clinical trials at the moment, being used as controlling obesity and stuff. Do you see that as a viable?
I think, yeah, it's going to, I think delivery is going to be difficult because Bace is so ubiquitously expressed. If you want it to penetrate the brain, you also have the difficulty of drugs passing through the blood-brain barrier. I do see, I mean, they have had quite a few attempts at making Bace1 inhibitors for Alzheimer's. I think the issue there is we still don't know what causes Alzheimer's, and it's not purely
It's not just amyloid.
So it's not down to amyloid. I think, yes, I think Bace1 inhibitors would be very useful. You just have to be able to titrate. Bace is obviously doing something very important if it's ubiquitously expressed, so you don't want to inhibit all of it.